I will be giving an introduction into the genomics platform RNA sequencing products. My talk is divided into two main sections based off of the two categories of RNA products that we have. The first is our more traditional bulk RNA sequencing, and the second is our single-cell RNA sequencing, which is a relatively newer technology. For each product, I'll be going into an overview of the lab workflow and the underlying molecular technology. I'll go over the inputs, outputs, and sequencing specifications, as well as some of the internal data QC that we perform. So first, we will go over our bulk RNA products. We have two. The first is TruSeq Strand-Specific mRNA. This is based off of a 3' poly-A capture and is generally used for higher quality RNA samples. The second is our Transcriptome capture. This is more of an exome-based targeting and is recommended for low quality RNA, such as highly degraded RNA or FFPE samples. So going right into our first product, TruSeq Stranded. And the first thing that happens when a sample comes through our door is an upfront QC step. We quantify the samples using RiboGreen, and we run them on a caliper to assess its quality. And I do have some slides later on going into a bit more on what this quality looks like and what we constitute a quote-unquote good RNA sample. But going into the process, we normalize our input to 100 to 400 nanograms of total RNA. We then perform mRNA purification using magnetic beads with thymine chains attached to their surface. This DT tail hybridizes to the 3' poly-A tail of mRNA molecules, you see on the left here. We can then purify this out of solution using magnets. From there, we go into first and second strand synthesis. Sorry, skipped ahead. First and second strand synthesis. The strand-specific protocol uses uracil in place of thymine here during the second strand synthesis. This comes into play when we go down to library construction and PCR amplification. This uracil actually quenches the polymerase during amplification, so it's this first strand that's getting amplified over and over again. And by doing this, we're able to preserve the original first strand of cDNA that corresponds directly to the original mRNA template. The TruSeq Stranded Kit is most often used for comparative transcriptomics, which measures the average expression level of genes across tissue samples, tumor samples, or cell lines. One of the biggest users of this product was GTEx, the Genotype Tissue Expression Project. This pretty much made up the first 2 or 3 years of my career. I just ran GTEx plates all the time. So what they did is they sequenced thousands of different tissue samples across many, many patients in order to create a transcriptome profile for all human tissue types. And this image from the side is actually from their portal. And if you're interested in exploring what they have a very nice portal. You can follow this link down here if you'd like to look into it a bit more. Moving on to our second product, transcriptome capture. So you can see that we actually accept a wider range of input, 100 nanograms to 1 microgram. However, this is optimized for low quality RNA, so we'll run whatever you give us, pretty much. As said previously, this is mostly for FFPE samples. An FFPE was a common tissue preservation method. It's great for preserving tissue structure and cell structure, not so great for preserving your DNA and RNA integrity. A lot of the time, these FFPE samples are historic samples. You're unable to go back and get more material, usually because the patient is already deceased. So a lot of the time when you're running this protocol, you're kind of just running what you have on hand and kind of hoping for the best. Now, the way that this works is it really skips that 3' capture. This is meant for highly degraded samples. This capture could pretty much deplete everything that you have. So instead, we go right into cDNA synthesis and create libraries using the Illumina library prep kit. From there, we actually go into a transcriptome capture. It's basically an RNA exome capture. We add probes that are specifically targeted to known mRNA-coded genes. And again, we can use magnetic separation to purify this out of the rest of our sample. The panel we currently use is called the Illumina Core Exome, which we refer to as ICE. It's a 38 megabase panel that targets 98.3% of the RefSeq exome. Here's a nice table comparing the inputs, outputs, and product specification for each of our bulk RNA products. For both products, we deliver demultiplex aggregated Picard BAM files. Our standard deliverable is 50 million reads aligned in pairs. However, for TruSeq Stranded, you do have the option for deeper coverage at 100 million reads in pairs. For batching samples together, we are a high throughput lab, so it is most cost effective to run high volume projects. We run everything on a 96 well plate, so we recommend batches of 94 to include your positive and negative controls, which we will include for you. We sequence TruSeq on the NovaSeq with 101 base paired reads, and TCAP on the HiSeq 2500 with 76 base paired reads. Turnaround time for both of these products, as well as all of our RNA products, actually are 6 to 8 weeks, usually shorter than that. And for both, we include data analysis and 5 years of storage. RNA extraction is available upon request for an additional cost. And for TruSeq, we also include ERCC controls, which are short RNA nucleotides that get a 2.5 microliter spike into each sample upfront. And this allows researchers to normalize their RNA data across different experiments or sample types. Here I included some quick guides on the RNA extraction process, as well as some of the recommended storage conditions for particular material types. Here you can find the preferred input amounts and the resulting RNA yield, as well as the kits that we use. As always, if you have any specific questions on extractions or storage, please email sampleslab@Broad or your project manager. So following extraction, there are two main quality scores that we look at. The first is RQS, which stands for RNA Quality Score. This is a metric we primarily use for our TruSeq Stranded protocol. It's on a scale of 1 to 10, 10 being the highest quality. It's calculated on a machine called the Caliper GX, which is somewhat similar to the Agilent BioA. And it takes an electropharogram, which you can see on the left, and it estimates the input amount and quality based on the areas between the peaks of the electropharogram. So from top to bottom, this is actually the same sample that is increasingly fragmented. And you can see how your peaks change as your RNA quality decreases. For FFPE sample, we use DV200 scores, which I show on the right. DV200 is simply the percentage of RNA under 200 nucleotides in length. And you can see again how your peaks shift as your RNA degrades. On the bottom right, you see a sample with a DV200 score of only 8%, and you can see that pretty much all of your fragments are under that blue 200 nucleotide range. So when running TCAP, we recommend using FFPE with a DV200 score of at least 30%. But again, we will take anything that you give us. It is important, so we will consider anything under that to be on risk. And when we say on risk, it basically means that you know that your input quality isn't great, but you want us to run it anyway and sort of just deliver whatever we can get off of the sequencer. Following our library construction protocols and sequencing, the data goes through the production RNA pipeline that is run and maintained by our data science platform. I'm not going to go too in-depth here since the subsequent primer sessions will, but essentially the RNA data goes through the Starliner and then the Picard pipeline to get us our aligned BAM and sequencing metrics. Internally, we review these metrics in our bi-weekly quality reporting meetings. Here's just a snippet of some of the recent SNETs we had go through TCAP on the left and TruSeq mRNA at the right. I'm just going to briefly go through some of the metrics that we look at to assess quality. The first is percent adapter. And high levels indicate a lot of adapter dimer that got amplified during your PCR reaction instead of your cDNA. This is usually indicative of low input quality. You just didn't have enough cDNA going into that PCR reaction to begin with. So the adapters hybridized to each other and those amplified instead. Reads aligned in pairs. As I said before, our deliverable is 50 million reads. If we don't hit that, we go back and rework the sample so it can get more reads on the sequencer. Percent mRNA just shows you how many of your reads are actually from mRNA molecules. It shows you how effective your poly-A capture was when running TruSeq, and it is also indicative of the quality of your starting material. The higher the percent mRNA usually means the higher quality of sample you had. 3' bias is essentially a measure of how many of your reads originated from the 3' end rather than the 5' end, and represents sort of the coverage that you have over the whole mRNA molecule. We only see this in TruSeq because we don't actually run the 3' poly-A capture in TCAT. And as I said earlier, poor quality RNA does degrade towards that 3' poly-A end. So when you have poor quality RNA going in, you're going to see a higher 3' bias. A nice little preview of what's to come. We are actually currently updating our TCAT protocol. Currently, we use Illumina library prep kits and the Illumina exome. We would like to switch to Kappa library prep, which includes UMIs, as well as to the Twist core exome. So we currently use Twist for our regular exomes and clinical exomes. So it makes sense for us to switch all of our exome products to the same panel. And UMIs are important because it allows us to better determine the origins of duplication. And if you've run RNA-seq before, you know that percent duplication is usually a problem. So we are interested in true biological duplicates that show us the actual relative expression levels. There are two other kinds of duplicates. PCR duplicates occur during the PCR amplification reaction. Optical duplicates arise during the actual sequencing. Basically, it happens where a single read structure on the flow cell is mistaken for multiple clusters. And by using UMIs, we'd be able to detect the origin of duplication and omit any non-biological duplicates from our data so that it can better represent the true biological transcriptome. And with that, we now move into the single cell world. We have two main products, sort of different flavors for each. The first is SmartSeq, the second is 10x. For SmartSeq, there are two main modes of running this. The first is true single cell, where you sort your samples into one cell per well of your plate. The second is population. You can do a small population of cells, 1,000 cells per well, or you can do ultra low input, 5 to 10 nanograms. This protocol obviously is ideal for very low input or low volume RNA samples. It is also ideal for low abundance or alternatively spliced transcripts, and it has been shown to have a higher sensitivity detecting more genes per cell than its 10x counterparts. In the case of population, this protocol is used to measure the distribution of expression levels, and the data resembles that of bulk RNA sequencing, but at much smaller inputs. When running one cell per well, you are able to look at the transcriptional changes at the single cell level, which can be used for studying regulatory networks and cellular responses, as well as identifying new cell types within a sample. The way this protocol works, so you first purify your RNA using RNA spray, and then you go into cDNA synthesis. And this uses a switching mechanism at the 5' end of the mRNA template, which is how SmartSeq gets its name. In this way, you're able to actually synthesize first and second strand within the same reaction. And the cDNA immediately undergoes amplification. And it is this improved reverse transcription and immediate cDNA amplification that enables the SmartSeq protocol to accept such low inputs. We then go into a tagmentation-based library construction, in which the DNA libraries are simultaneously fragmented and primed with the appropriate sequencing adapters and indices, as you can see on the right. For SmartSeq input, we require your cells to already be sorted and lysed. We do require a specific low bind plate that you have to purchase in order to prevent loss of RNA. We don't recommend using any other plate besides this. We've had some people try to use their own plates in the past and they end up losing all of their starting material. So please order these plates. Sequencing is done on walk-up. We don't have a production analysis pipeline yet. So we deliver demultiplexed FASTQs. And if you've used walk-up in the past, you're familiar with our get sites. It's basically just a site where you can download your data. We provide 30 days of storage, and you will get a reminder email when your get site is about to expire. We do strongly recommend that you download it in the first 30 days because it will get automatically deleted. For batching, again, we run everything in a 96-well plate. If you want to include controls, that is up to you. If you have less than 96 samples, that is also completely fine. However, we do charge on a per plate basis. Sequencing is done on a NextSeq. When you're running single cell, you can actually put 4 plates onto a single flow cell to get about 1 million reads per cell, which is more than enough coverage. Otherwise, it's about 3 million reads per well. As I said, we don't run a full pipeline, but we do run internal data QCs on the sequencing run and the sample quality. So this is another Tableau report. Different colors here represent different cell types, and you can see how different cell types perform differently through this protocol. A couple of the metrics that we look at are total genes detected, total reads, percent alignment, percent mitochondrial RNA, and percent mRNA. When we look at percent mitochondrial, high levels actually indicate high levels of cell death. When the cell dies, it'll release its mitochondrial RNA. And since this is actually a different transcriptome than the rest of our RNA, we don't actually want to sequence this, not for disease studies at least. This is just another quick graph of some of our metrics. Bottom left is, the left is percent mitochondrial versus total reads. And you can see that a lot of our high percent mitochondrial corresponds to very low reads, which usually means that you just didn't have a lot going into the process to begin with. Again, usually indicative of cell death. On the right is genes detected as a function of total reads. And you can see it's almost a direct positive correlation, but once you get to a certain level, you sort of reach a sequencing saturation, where as you add on more reads, you're not really increasing your detection power by that much. And that is why we do recommend putting 4 plates of single cell onto one flow cell, because 1 million reads per cell is more than enough coverage, and past that, you do hit that sequencing saturation. And moving on to our shiny newest product called 10X. The way this technology works is it takes dissociated cell suspension, but not lice. These are alive cells, as alive as you can get them at least. So they have, it's called a chromium box. It's honestly, it's a small box, and you load your sample onto a chip, and the chromium machine uses microfluidics to isolate single cells into oil emulsion droplets. These also contain a special bead, which 10X called gem beads, gel bead in emulsion droplet bead. It's very creative. So these beads are functionalized to capture the mRNA within the cell, and every bead has a unique 10X barcode. So all mRNA molecules within the cell will have the same 10X barcode, and this will allow us to demultiplex down to the single cell level downstream. The benefits of this technology, it allows you to take a tissue or tumor sample and study the full heterogeneity of the cells within the sample in order to characterize the different cell populations and identify novel cell types. There are 2 main gene expression protocols. They follow more or less this, well, actually pretty much the same lab workflow. And they differ in the sense of which end of the mRNA strand they capture. So there's 3' and 5'. The 3' has that typical poly-DT tail for that polyase selection. 5' has that switch oligo that we saw in SmartSeq. So the input for these protocols are cell suspension pretty much as soon as it's dissociated. You count it and load it onto your chip to create the single cell droplets. The cells within the droplets are lysed to release the mRNA that then bind to the beads and get barcoded with that 10x barcode. You then perform reverse transcription to make cDNA. You amplify the cDNA and then you proceed to library construction. And we do have a couple of add-ons for each of our expression protocols. The first is for 3'. This is called cell hashing. And this is used to multiplex multiple experiments into a single 10x channel. A couple of reasons for doing this. You can mitigate batch effects between running different channels, reducing costs, that's probably the big one. And then it also allows for doublet detection, which the 10x pipeline does detect this somewhat based on UMI counts, but it doesn't always catch all of them. And this actually helps you omit even more doublets that you don't want. And when I say doublet, I mean there's 2 cells captured into 1 droplet. So the way that this pipeline works is when we reach the cDNA cleanup step, your hashtag, oh, sorry, I'll start from the beginning. So you stain each of your samples with custom barcoded antibodies, and you can multiplex them together because now that they're tagged with these certain antibodies, you make libraries downstream of these antibody sequences. So this is following cDNA cleanup. These hashtag libraries are smaller and will be in the supernatant of your cDNA cleanup. So in this case, you actually save your supernatant and you pretty much put it right through SPRI cleanups and PCR to amplify it. You then spike it back into your main library, sequence it as usual, and then there's a pipeline under Cumulus, which is owned and maintained by the KCO, where you can demultiplex based on these original sequences. For 5', our main use for 5' is actually immune cell profiling. Now 5' beads have a capture sequence specifically for the antibodies found on T and B cells. This is done with 5' because the VDJ variable region of an antibody is located on the 5' end as well. So this follows a similar lab workflow. Instead of the supernatant, you actually just take an aliquot of your cDNA library. You enrich for T or B cells, and then you go through library construction and PCR, and you spike it back into your library again, which is optional. You can sequence them separately, but you will be getting a lot of reads for relatively small libraries. This is just a quick preview of what the analysis pipeline looks like. I'm not going to go too in depth. As I said, the rest of the talks will go into a lot more detail on this. But I just wanted to point out the 10x analysis pipeline is called Cellranger. Basically, the first step is a make FASTQ step. It's really just a wrapper of the Illumina demultiplex. And then it goes through the count protocol, where it demultiplexes back down to the single cell level using the 10x barcodes. And for immune profiling, there is a different pipeline for your VDJ libraries. And they actually have a nice software called the Loop Cell Browser, and you can plug your data from your VDJ and your gene expression analysis to perform integrated analysis. So you can look at both your immune profile as well as your expression. I just wanted to give you a quick introduction of what your Cellranger outputs will look like when you run it. So it first spits out a basic HTML summary page. It gives you the number of cells you recovered, the reads per cell, and the genes detected per cell. On the bottom left, there's a barcode rank plot. It plots the UMI counts on the y-axis versus barcodes on the x-axis. So a steep drop-off that you can see here shows good separation between what Cellranger is counting real cells in dark blue versus, versus background noise in gray. And here background noise would mostly just be from empty droplets that don't contain any cell. So naturally, we want to omit those from our data. On the right, you can see genes per cell as a function of reads per cell. Again, you see a positive correlation, but once you get to a certain amount, you do start hitting a sequencing saturation. Cellranger also performs a secondary analysis, which will perform auto-clustering based on UMI counts and expression profiles. You are able to control how many clusters you want, and if you use their Loop browser software, you can search for specific genes, visualize the expression levels of that gene across your different clusters, and in that way you can identify specific cell types or new cell types. Some input specifications for 10x. Both protocols take either single cells or even isolated nuclei. We do require cell suspension that has already been dissociated, and in the case of hashing, already stained and multiplexed beforehand. We deliver raw BCLs through a get site, and Cellranger is available upon request through our tag analysis team. There is no minimum number of channels to run. However, you do have to buy a full flow cell of walk-up. And for hashing, the most that you can do recommended into a sample is 8 samples per channel. Again, we do walk-up sequencing. You have the choice between a NextSeq, HiSeq, or NovaSeq. And when deciding on what kind of sequencing you want, so we actually have a couple of handy-dandy calculators to figure out what sequencer best fits your need. So the first thing you want to know, you should, starting your project, you'll want to know how many cells you want to recover to begin with and sort of the target reads per cell. A lot of people do 20, 50, or 100,000 reads per cell. So in the sense of this, for this calculator, you would just plug in your recovery, target reads per cell, and you just click whether or not you're making a T or a B cell library. It'll automatically calculate total reads per sample, and then it plugs this into how many reads per flow cell you get and how many samples you'll be able to fit onto that flow cell. So if you're running an S4, you would put 8 channels onto one flow cell. Cell count is important to know before loading. There's a 60% encapsulation rate on that Chromium box. So you want to make sure you're loading enough. But you also want to make sure you're not loading too much, because overloading can increase your doublet rates, and it also risks clogging the machine, which 10x will not refund us for, and it's very expensive. We don't offer analysis as our standard product offering. However, it is available upon request. And we do not perform tissue dissociation ourselves. However, we strategically put our lab space in a flow core lab, and they offer all of the equipment and lab space you need to run it on your own. It's sort of a book for fee. And if you're interested in running this, contact their lab director, Patricia Rogers. She goes by Trisha. And this is actually very beneficial because you can just dissociate your cells right at the Broad. You don't need to buy any of your own equipment. And then as soon as you're done dissociating and counting, you just walk to the back of that lab and hand it off to our 10x lab techs, and they will run it for you immediately. And you don't have to worry about time between dissociation and your cells dying. So yeah, it's worked out pretty well so far. And that concludes my talk. If you have any questions or feedback, now's the time to ask them, or you can email either one of these.
Great, thank you so much, Carolyn. That was a wonderful set of information about all of the various products that are available. The technology is evolving so rapidly.
Yes, it is.
We've already had 2 questions, and then we'll also keep an eye out for raised hands. So I'll just read the first question from Matthew Warman, which asked about whether you could speak a little bit about fixed versus fresh cells for the 10x platform, and whether or not it's possible to create your own capture sequencing for the 5' type of products. In particular, they're looking for transcripts with a mutation near the 5' end that might be somatic instead of germline. Okay.
I might let Cole take this one over.
Yeah, sure.
Lab operations manager. He's good with the technical stuff.
So fixation, there's 2 kind of ways you can kind of go about that. Tissue fixation, we have not had a lot of success in that. If you have frozen tissue, fixed tissue, and you really want to stick on the kind of that tissue side, every time we've done it, just too much cell death, too much mitochondrial, we recommend that you run nuclei through the platform. It's really just designed for that. Methanol fixation of cells, a little bit different story. You can actually recover live cells from that and get a little bit better results. And it's actually, there's a pre-approved process from 10x about rehydration and everything like that, that they actually approve for use for their system, which has been shown to work. We don't run a lot of it, but most of the time that we get frozen or fixed tissue, you kind of have to go nuclei. And what's the other one?
Just whether it's customized 5' capture to look at somatic versus germline variants.
Right. So if you wanted to do something like that, I think like Carolyn said, we kind of use the TotalSeq A and B kind of pre-made BioLegend antibodies. You can go through BioLegend and create your own sequence. This would be a process called feature barcoding. So if you just want to pull from that capture sequence, you can determine what that capture sequence really is. And BioLegend, that's what their whole TotalSeq process is about. And then you would make your customized panel based off of that. Obviously, for ease of use, we use kind of a pre-made panel that they sell.
And have you used those custom products much with success?
We have never needed to make a custom panel.
Next, there was a hand raise from Steve Alexander. Would you like to voice your question, Steve?
When you look at the T-cell repertoires, what's the best software that you're using for looking at the T-cell repertoires that come out of the data?
What software? We use the vLoop browser software for immune profiling. Is that your question?
Yeah, that's right.
Perfect. And if anyone else has experience with T cells and would like to add to the Q&A, any thoughts about that too? I know it's a place where there's a lot of fast-moving software development to be able to enable very specified analysis. There's another question from Hun-Sik Yang, which was, which library would you recommend for low volume samples? For example, monocytes at less than 10 to the 6th from less than 10 cc's of blood.
Can I ask how much RNA would come from 10 to the -6 monocytes? Like, would you expect?
That's something I don't have good background on.
For that question, like 10 to the 6 monocytes, 10 cc of blood. So that's probably what, like 100 cells per mic, give or take. Like that's 1 million cells for 100 cc's is like 10,000 mics. So I would probably recommend SmartSeq. TruSeq would be really kind of pushing that kind of input. You can decide whether or not you'd want to go population or kind of a higher single cell. You can put like 100 cells in and then still run it through the single cell pipeline just to get kind of more quality data out, because you might not really have enough to even run the 1,000 to 10,000 cells per well. SmartSeq though.
Thank you. And And then just to respond to a question in the chat, we will definitely have this primer, which thanks to Carolyn, Carrie, and Cole will be recorded. And so this will be posted to the both internal Broad as well as to the external YouTube. So we'll make sure to make that available to the community. So feel free to either raise your hand or type any more questions in the chat. One of my questions was, what do you think is the ideal time for a group to approach you in terms of designing an experiment? And are there any particular pitfalls that you most commonly see when there's kind of limiting samples or other concerns for sample analysis?
In terms of like approach, we're pretty much open all the time to take on kind of new projects. Kind of the bigger issues we see is, especially in the single cell world, a lot less on the RNA side, is keeping cells alive, and that kind of quick turnaround time between cell dissociation, if they have to do anything with the tissue up front or anything like that, into chip loading, especially if we have to add, they need to add like hashing or any kind of antibodies up front for looking for kind of custom areas. So that's kind of been our biggest kind of hurdle that we kind of have to get around, is kind of planning it out, especially in this kind of time where we can't have as many people in the lab, kind of working with the flow core, making sure that everybody can share space. And also just like people's comfort level in dissociating those kind of tissues. Some people would expect us to be able to do it, but since we run, 150 channels a month, we just don't have that wide range of expertise at the moment with the staff to be able to handle various cell types, to be able to dissociate them all. So just kind of making sure that they understand everything that needs to go in to running single cell is probably like kind of the biggest challenge, and trying to make that as clear as possible and setting the guidelines to give them the best chance of success, because we do know it is an expensive product. So it's really, we don't want to kind of ruin anyone's like runs in the beginning. And we always recommend a pilot phase. So don't cannonball in.
Thank you. There's another question from Matt Mormon in the Q&A, which is regarding their studies of mouse cartilage, where they get about 1,000 total cells as a yield from an experiment. To optimize RNA retention, are there any special tubes that are low adhesion or have other properties that are good to think of in kind of those upstream sample collection steps?
Yes. So most companies do make kind of those low bind tubes, because what we do see in a lot of areas is material gets stuck to the sides of the tubes. And if you're dealing with really, really kind of low amounts, we always recommend, I know Carolyn put in like the catalog number of what we recommend for just the plate, just to get the most out of it, because we do go down to like 10 picograms of RNA. So even losing half of that would be dramatic. So yeah, they do have special things. There's also low retention tips if you want to work by hand. Rainin makes great low retention tips if you want to use your multi-channel and do it on your lab bench.